Hi, I'm Karin Dumstrøy, and I'm from the EMBO Journal, and I have the great pleasure of talking with Pamela Bjorkman here at the 78th Cold Spring Harbor Symposium, which this year is on the topic of immunity and tolerance. Thank you for taking the time.
Thank you.
So your recent work, and this is also what you talked about a few days ago, was on understanding and sort of optimizing how antibodies will bind and neutralize the HIV virus.
Yes.
So before we get into the sort of the specifics of that, can you maybe sort of briefly outline sort of what's the normal immune response if a host will see, or if I will see, my system will see a pathogen?
Okay, well, to answer that, I'm going to ignore cellular mediated responses for the, so I'll just focus on antibodies. So if you have a normal looking virus, say an enveloped virus like influenza virus, then it has spike proteins that are there to help it get inside the cell.
And so these are literally spikes that sort of project from
well, people call them spikes. They're just proteins.
Yeah.
They often have a spike-like shape, but flu hemagglutinin, for example, is a spike-like shape. And so on influenza virus, it's about the same size as HIV, so about 100 to 150 angstroms in diameter.
Okay.
And it has 440 spikes on its surface.
On the surface, yeah.
And so the spikes are quite close together, and antibodies, when they bind, of course, antibodies have two identical arms. And so the idea is that when antibodies come in and neutralize a flu virus, they would be binding bivalently, so like this.
So with their two arms?
With both arms. And I mean, antibodies, I'm sure, evolved to have two identical arms so that they could bind with avidity. So in that case, you know, anything binds, any chemical reaction is in equilibrium, and so you have a certain on rate and off rate. So at a certain level, the one arm is falling off, and if both arms are binding, then the antibody will likely remain attached to the virus. So basically the avidity of the interaction, which is caused by having two identical arms that can bind at the same time, causes the off rate to slow down a lot.
So it will stay attached to
it should stay attached for a certain amount of time, probably long enough to neutralize the virus or recruit effector cells through its Fc or whatever. Okay, so that's the way it should work. And if you look at what viruses look like for viruses to which we either have a vaccine that raises antibodies or your own immune system raises effective antibodies, they generally have a dense array of spikes on their surface. Now you look at HIV, and I thought it was remarkable when the first cryo-EM images came out in 2006 from Kenneth Rue's lab and then later from Sriran Subramaniam's lab. It was really clear that HIV just looks different. So their pictures look like this bald sphere, and it has, on average, 14 spikes. So, you know, the number varies, 10 to 30 or whatever. And this was known from earlier biochemical work. So it's not just the cryo-EM studies, it's a number of studies. So hardly any spikes. And so we started looking at these things, and it kind of explained why our kind of naive ideas about how to improve HIV antibodies weren't working.
Yeah.
So we were trying to increase the valency and get them to bind with better avidity. And then we started noticing, well, you know, a normal IgG probably doesn't bind with avidity to HIV. So where it could bind like that to flu, it's going to bind like that with one arm most of the time. Most of the time. I mean, you know, some statistical amount of times it might be able to reach between spikes because they're different on every virus and so on. So in thinking about this, we thought, well, this is another reason that HIV is able to evade antibodies successfully. And the idea would be that, you know, when you raise the first antibody to a particular virus, it binds tightly. And even though it has one arm, it doesn't really matter. And then HIV starts to mutate. And then that one arm, its off rate increases a lot, and it just falls off.
So it falls off, and so it can't stay.
Yeah, so if it had two arms binding, it should be able to tolerate mutations more. So the way I like to describe it is that the combination of a rapid mutation rate in HIV and predominantly monovalent binding is part of the reason that HIV is so successful and our immune system doesn't do a great job.
So some patients develop these,
Yeah.
what they're termed broadly neutralizing antibodies.
Yes.
And I think a lot has been learned from how they
Yes.
look. So, so what do these broadly neutralizing antibodies look like? And what is so
They
unique
look-
about them?
Well, they have longer complementarity determining regions
Okay.
that can sometimes reach through the glycan shield. They have a variety of different mechanisms. They're more highly somatically mutated than normal antibodies. And so they, we and others study how they bind to HIV spikes, usually, usually by crystallography to the GP120 portion of the spike. And so they do bind.
And how do they bind better?
Well, they bind, they have various ways of increasing their monovalent affinity, and they bind to conserved regions that the virus has trouble mutating against.
Okay.
Now, that being said, I think that most of those are binding usually monovalently.
Okay, so still the one-arm
Yes,
binding, yeah?
and if you look at the particular person from whom those broadly neutralizing antibodies were isolated, that person's viruses are not neutralized by them. The reason they're broadly neutralizing is that in a panel of viruses isolated from lots of different people, they tend to get, like, say, 90% of those. But that person's viruses are already resistant.
Yeah. So how do you use this to sort of optimize and generate antibodies that could be used in therapy?
Yeah.
And I presume in preventive vaccines as well at
Yes.
some point, yeah?
Preventive vaccines is a bit harder. You have to learn how to elicit them. And
Yeah.
because they have so many somatic mutations, that's hard.
Yeah.
So that's,
So let's say therapy for the
Okay,
moment,
so
yeah.
let's say therapy. So you could inject them or you could deliver them by gene therapy.
Yeah.
And many people are working on this, and it will someday probably be a practical reality. Okay, so we try to improve them two ways. One of them is just structure-based rational design, and then we're just saying, okay, it binds monovalently, we can't help that. But we improve the actual interface. And so we go in and look at X-ray crystal structures of antibodies bound to one of the arms, and we say, oh, well, I'll bet if we changed this residue on the antibody, it would bind better.
Okay.
And there are computational ways to do this as well that we're doing. So some of it's visual inspection, some of it's computational. And so we've made some improvements to Michelle Nussenswag's antibodies, which are already very potent and very broad, as isolated from the people he's isolated them from. So that's one way of doing it. And that's sort of the rational way. And the sort of irrational or less conventional way we're trying to do it is to just take antibodies, whether they're improved or not doesn't really matter, and link them together in such a way that they would bind at the same time to the viral spike, to one viral spike.
So you're making more arms in a way, yeah?
You could do it that way, yes.
Yeah.
Yes. It doesn't have to be more arms, though. What we're trying to do is so if you have spikes and they're too far apart to bind bivalently between spikes, so inter-spike cross-linking, if what you could do is bind within the same spike, intra-spike
I see, yeah.
cross-linking, then the distance between them doesn't matter.
Yeah.
So the distance between them is going to be wildly different from one virus to the next. You can never get anything that would work for all HIV, even all HIV of one strain.
Yeah.
Okay, so the idea is you have this trimer, and if you could design something that would bind to site A and site B at the same time on each individual monomer, then that would give you synergistic binding. And I'm not talking about it binds to A or B.
No, it's both.
Those are very easy. It's very
Yeah.
easy to link antibodies and get it to bind to site A and on some and site B on the other. We're talking about binding at the same time. And so that requires either designing it rationally by looking at the structures. That's a bit hard because our structural knowledge of the HIV spike trimer is incomplete. We have structures of truncated monomers only. So you can sort of try to design your way through this, but it's difficult. But we've had some success in making libraries of HIV binding reagents. We call them widgets, so we have, say, 30 of these. And then we link them with variable lengths of double-stranded DNA, which serves as kind of a stiff ruler. So we figure out which combination of two widgets works and what length of linker of DNA is between them. And of course, DNA is 3.4 angstroms per turn, so we sort of know what the distance is. And then once we get that, and so we've succeeded in getting things that act synergistically. That is, to site A and site B, our data suggested they're binding at the same time, synergistically, so they're better than A alone or B alone, or the combination of A and B not covalently linked.
And I guess these antibodies should be, if the virus mutates, they should be sort of...
Well, that's the hope, is that, so the virus, highly active antiretroviral therapy works because you have, say, 3 of them, and the virus can't mutate fast enough to mutate against 3 things. Okay, so if you had, say, 2 or 3 sites, but even 2, I think it would work with even 2. What would happen is the virus would have to put mutations in site A and site B, which it might not be able to do. But in addition, those mutations wouldn't work very well because of the avidity effect. So if you've got the thing binding at the same time and you put a mutation in site A, then it might be a low affinity interaction, but it's being tethered there by
still
site
has another
B.
anchor,
And so I think that avidity can compensate for low affinity. So avidity would serve as a buffer against the mutation. So it could mutate maybe even in site A and site B. And then if you could, so you could make that, let's say. So we have leads on how to make that. But now what you do is you either dimerize that with an antibody FC, so it's binding to 2 of the 3 subunits in the trimer, and so it would bind to 4 sites instead of 2. And then you'd have FC effector functions and increased half-life and so on. Or you could trimerize it with a synthetic trimerization domain, and then you'd have it binding to 6 sites. And I cannot imagine that HIV could mutate to get around that. I don't think it could.
Well, so it
remains
sounds like
to be seen.
What did you call the unrational approach? Seems like very promising, yeah?
Promising but difficult. But it's something that I think even if it doesn't work to make a therapeutic, I mean, at worst, we will learn about how HIV mutates and what's important and sort of how to neutralize it. We'll learn about neutralization mechanisms. And at best, we could end up with a therapeutic that could be delivered.
What is the missing structure in the HIV virus field?
It's the spike trimer. Because, like I said, we have structures from several labs of various GP120 subunits, which are monomeric. And then those are truncated because it has flexible loops that will not let you crystallize the protein if you include them. So the loops are truncated in various ways. And then we don't really understand how the thing trimerizes. There are cryo-EM structures, single particle cryo-EM, and also electron tomography cryo-EM that give us some idea what the trimer looks like. But again, the loops are missing in these structures because they're flexible. So I'd say understanding what the trimer looks like is just not very well understood, and it's proving very difficult. You see, I came from Don Wiley's lab as a graduate student, and in 1981, he and Ian Wilson's published the structure of flu virus hemagglutinin, a spike trimer on flu. And it's just not happening for HIV spike trimers. It's just more difficult.
So where do you see your research going within the next 5-10 years?
Well,
what would you love to have the answers to?
I would love to have this reagent. I would love to have a series of these reagents, and then I would like to participate in a clinical trial to see if it was effective at preventing HIV acquisition or therapeutic in people who are already infected with HIV. And there's various ways you could imagine this could be done. So what I would really like to do is be involved in repeats of experiments that were done, say, 5-10 years ago, where in humans, where people were taken off. So people were on antiretroviral drugs, their viral load goes down. But then as soon as you take them off, the viral load goes back up. That's well known. And so what was done by several different groups was to give them neutralizing antibodies after taking them off highly active antiretroviral therapy. And the antibodies delayed the viral rebound, but they did not prevent it.
Yeah.
But those papers were published in 2005 and 2007 before people had the kind of antibodies they have now. And I would just love to see this repeated with the broadly neutralizing antibodies in their conventional forms, because they're 100 to 1,000-fold more potent, and some of them are more broad. Okay, so in their conventional forms, and then testing if something designed with an alternative architecture could be perhaps more effective at delaying or just eliminating viral rebound. And so this is going to take a lot of work, but a huge amount will be learned from these sorts of studies.
Yeah.
And I'm not minimizing the difficulty in any form of unnatural antibody is potential immune responses against the parts that are unnatural.
Yeah.
And so, of course, we're using human proteins as linkers between subunits and so on. But I mean, the only way of knowing if those are going to be immunogenic, unfortunately,
is in
clinical trials for efficacy. I mean, not efficacy, for safety.
For safety, yeah.
And I realize this is something we can't predict, and we can't use mouse or non-human primate models to answer this question. But I'm hoping it won't really matter, because if you're delivering a drug like, let's say, a protein drug like erythropoietin, and you mount an immune response against it because it's slightly unnatural, you're going to die, right? Because you have to have EPO.
Yeah.
But if you're mounting an immune response to an antibody that's being delivered therapeutically, you'll make it not work as well.
Yeah.
But I hope that it would not be dangerous.
Yeah.
But it remains to be seen if most people would mount immune responses, and if so, would it just reduce the efficacy?
Yeah. Well, thank you very much for taking the time.
Okay. Thank you.